Hello and welcome to this presentation. My name is Arne Materna. I'm a member of the Board of Directors and co-CEO of Biome. And over the course of the next 20 minutes, we shall discuss different technologies for routine microbiome testing in the context of women's health. I would like to thank Thermo Fisher Scientific for the opportunity to present to you today. The human microbiome is an integral part of our physiology. It's been associated with numerous health and disease conditions, and ever since the end of the Human Microbiome Project, which concluded in 2017, I believe, research into the microbiome has continued to shape and inform our understanding of clinical and environmental microbiology. The microbiome also has ever since been a steady target of biomedical research. Now, historically, the focus of this research has been on the intestinal or gut microbiome. However, microbiome has evolved and moved to include other body sites, such as the urogenital and skin microbiomes, which have been associated with numerous skin conditions, vaginal infections, and reproductive health. Unlike the commonly studied fecal specimen, which essentially to 95% is comprised of bacterial biomass, skin and vaginal specimen can be characterized by very scarce amounts of microbial biomass. At the same time, these samples tend to be heavily contaminated with high amounts of human DNA background. This situation proves challenging for sequencing-based methods, and it adversely affects the sensitivity, reproducibility, and quantitative accuracy of these methods, all the while driving up costs. At Biome, the vaginal microbiome has entered firmly the center of our attention. Unlike other body sites, the vaginal microbiome can be characterized by well-defined and conserved patterns that have been first described as community state types, or CST for short, by Jacques Ravel and co-workers. Since then, CSTs have been confirmed to be conserved among reproductive aged women and across ethnicities and geographies in numerous studies, including large population studies. A healthy vaginal microbiome in homeostasis is usually dominated by one of several lactobacillus species, with Lactobacillus crispatus being the most prevalent one and characteristic of community state type or CST1. Others include Lactobacillus gasseri, Lactobacillus jansenii, or Lactobacillus iners, which is characteristic of CST3, an unstable state. The interaction of these beneficial lactobacilli and their host is fairly well understood. These lactobacillus species manage to evade the innate immune response of their host and acidify the vaginal environment. Lowering the vaginal pH helps prevent overgrowth with commensals and the colonization with opportunistic pathogens. If the vaginal microbiome shifts from healthy homeostasis to dysbiosis, we typically see the emergence of patterns that are summarized as CST4. And characteristic of CST4 or dysbiosis is the increase of microbial diversity and the colonization with anaerobic but also aerobic pathogens, including Gardnerella vaginalis, Prevotella species, but also Klebsiella and Streptococcus species. Also characteristic of dysbiosis is again the increase of the vaginal pH and the activity of enzymes such as sialidases that disrupt epithelial membranes and thereby contribute to the etiology of certain infectious conditions, including bacterial vaginosis and vaginitis. Also, the risk for sexually transmitted infections, including HIV, is significantly increased, and dysbiosis contributes to adverse pregnancy outcomes, including preterm birth and pregnancy loss. At Biome, we therefore believe that routine testing of the vaginal microbiome presents an opportunity to advance women's health. Consider the example of reproductive health shown here. Now, 1 in 6 couples are struggling with infertility. Fertility treatments help, but at success rates of 30%, failure is more likely than success. And unbeknownst to many physicians and patients, for as many as 1 in 3 women, the failure rates can be as high as 92%, due to microbiological vaginal conditions such as microbiome dysbiosis, BV, AV, and others that are very prevalent, highly recurrent, potentially treatable, yet in these settings often underdiagnosed. If you then consider the health economic impact, but also the psychological trauma that stems from treatment failure and adverse pregnancy outcomes, then it becomes clear why BioMe embraces the vision to develop a single molecular test to detect all vaginal microbiological risk factors and to inform physicians accordingly and allow them to treat and respond, but also to empower women to be aware and potentially better time reproductive treatments for a greater chance at motherhood. To this end, we have developed, tested, and launched a research use panel targeting vaginal health. Now, for the remainder of the presentation, I shall shift gears and focus on the technology that we have selected for this panel and the benefits that we believe this technology to provide. When we've set out to develop a routine vaginal microbiome test technology, we engaged with experts to understand which key criteria does a vaginal or does a microbiome technology in general have to fulfill in order to be suitable for skin and vaginal microbiome testing. And we were able to identify 6 requirements. The first is a requirement for multi-species detection, which is a quite obvious requirement for any method used to characterize microbial community composition. The second is the taxonomic resolution at subspecies level. The third requirement is a requirement of the platform to be robust and perform robustly when faced with low bacterial biomass and high human DNA contamination of the sample. The fourth requirement is a requirement to be able to absolutely quantify microbial loads. Relative abundance measurements are not enough, and I will later argue why. Finally, turnaround time and cost must be conducive to routine testing and a timely decision making. BioMe decided to partner with Thermo Fisher Scientific for the development of our method for precision microbiome profiling, or PMP for short. PMP relies on a highly parallelized, miniaturized approach to singleplex quantitative PCR. PMP thereby uses very small nanoliter reaction volumes, which can be individually coded with taxon or species-specific molecular primers and probes. These reaction volumes provide the superior performance of singleplex quantitative PCR and reduce reagent crosstalk. These volumes also can be grouped in so-called subarrays, which enables us to simultaneously detect any number of bacterial targets in multiples of 56. And finally, these subarrays can be grouped into a large consumable, which allows us to analyze dozens of samples in the same analysis run, essentially. These consumables for our PMP test are manufactured exclusively for BioMe and to the highest standards by our partners at Thermo Fisher Scientific. But back to the original 6 requirements. During the development, we engaged in multiple studies to ensure that the PMP technology is able to address the requirements that I've mentioned earlier. And we started with an assessment for multi-species detection and taxonomic resolution in collaboration with Professor Lars Engstrand's group at Karolinska in Sweden. Together we decided to compare the performance of PMP to the gold standard for microbiome analysis, referred to as shotgun metagenomic sequencing. We also included 16S sequencing for comparison. We further decided to maximize the challenge to PMP, and in doing so, we developed a 110 species panel and targeted fecal microbiome samples, as the gut microbiome is highly diverse. Now, what you can see here on the left-hand side is a chart that compares the different platforms across 74 fecal samples for their capability to accurately reproduce microbial community composition. The chart on the left side is a so-called principal coordinate analysis plot that seeks to maximize the difference between samples based on compositional dissimilarities. We were very pleased to see that the gold standard shotgun in blue and PMP in green here are highly concordant. 16S slightly struggles to reproduce microbial communities, which however is to be expected in this diverse sample type. Moving on to the chart in the middle, demonstrates the high concordance between PMP and the gold standard when comparing relative taxon abundances. We were very pleased to see correlation coefficients in the 90s. And finally, moving on to the right-hand chart, where we've compared 16S to the gold standard, seeing less concordance caused by slightly elevated miss rates of 16S in these samples. Now, after having established that PMP is suitable for the profiling of even complex microbial communities with highest species level, highest taxonomic resolution to the subspecies level, we wanted to evaluate the performance of PMP when met with samples that are very low in microbial biomass and contaminated with human DNA background. To that end, we partnered with a group of Professor Lieberman at MIT. And once again, we decided to challenge PMP maximally by selecting samples from from the skin of the lower leg. Now, this is a specimen that is notorious for very low microbial biomass and a high amount of human DNA background contamination from shed skin cells. We compared once again across the three methods, PMP, shotgun as the gold standard, and 16S, the ability of the methods to reproduce microbial composition. Here represented in the form of a stacked bar chart on the left-hand side with different colors representing different microbial genera. We can see that PMP and the gold standard are very concordant, while 16S with this particular specimen type essentially misses entire genera while overrepresenting QT bacterium. The chart in the middle quantifies this measurement to a better extent. Shown again here is a measurement of microbial community composition or beta diversity. Specifically, we chose the Bray-Curtis dissimilarity metric. And we compared different platforms pairwise to each other. Now, a lower dissimilarity means higher concordance between platforms, so lower is better. And once again, we were very pleased by seeing a strong concordance between shotgun versus PMP, shown here in the middle. However, it was remarkable how drastic and significant the underperformance was, and in other words, the discordance with the gold standard of 16S when applied with these across these 43 skin samples. We saw a vastly different compositional profile between shotgun and 16S, and also between PMP and 16S. The chart on the right-hand side to some extent sheds some light as to why 16S underperforms. Here we plotted alpha diversity, which is a measure of microbiome richness and evenness. And in our case, this metric is relevant as it compares the method's ability to detect taxa and to accurately determine relative taxon abundances. Once again, we see strong concordance between PMP and the gold standard shotgun metagenomic sequencing. If anything, then the tighter distribution of data for PMP suggests greater reproducibility. However, we see that the underperformance of 16S is not only due to a very high miss rate, but also stems from essentially the inability to return any meaningful abundance measurements. This is one of several recent studies that actually draws into question if 16S sequencing is a valid method for the analysis of skin microbiome samples. But back to our method PMP. After establishing the strong performance with challenging skin and skin-like specimen, we tackled an issue that is very challenging to accomplish with sequencing in general, regardless of the sequencing approach. The absolute quantification of bacterial and fungal loads. And as promised earlier, I shall now provide you with an example for why absolute abundance measurements are important in the context of women's health and skin health. To that effect, I'll take you on a short detour into the clinical development of therapeutics that target vaginal infections. GIDEA Biotech is a microbiome drug developer advancing a new drug candidate called PHIVE along clinical stage development. P-Hive has a unique dual mode of action. It lowers and restores the vaginal pH while introducing a carbohydrate substrate that disrupts biofilms often associated with antibiotic-resistant infections. One of the main reasons for GIDEA selecting our PMP technology for their phase 2b clinical trials is PMP's ability to return accurate absolute abundance measurements. The community is becoming increasingly aware that relative abundance measurements can contribute to a phenomenon called compositionality, which in turn can lead to false positive discoveries. And allow me to provide you on the lower right-hand side a chart that I'll use to explain this phenomenon of compositionality. So what you see there are abundance time series, as they are common for before versus after intervention studies. The lines in black represent relative abundance measurements as returned by sequencing. The lines in color correspond to absolute abundance measurements. And now consider example A, in which you are, for example, the developer of a live biotherapeutic, such as a probiotic strain or consortium, and you're introducing this probiotic into the host. Now the probiotic does what it is supposed to. It engrafts in the host environment, and as a consequence, the relative abundance of the probiotic strain or consortium increases, but also in absolute terms, the abundance increases, contributing to an increase in overall total bacterial load or biomass in the host. Now, next, you'd like to establish the efficacy of the probiotic to displace the here disease-causative pathogen. And to that end, you measure again abundance of the pathogen over time. And in relative terms, the abundance of the pathogen increases. But in our scenario, this decrease is due to the fact that the overall microbial load has increased during engraftment of the probiotic strain or consortium. Now, in absolute abundance measurement terms, however, the colonization with the pathogen remains unchanged, as strong as before, and so does the pathogenic effect that this pathogen exerts on the patient. Now, sequencing and relative abundance measurements would false positively suggest an efficacious displacement of the pathogen. And this is a mistake that potentially is not only expensive to a drug developer, but of course also to patients. I'm pleased that I can share with you some preliminary study results from our partners at GIDEA. On the right-hand side, you see 4 endpoints, and it's important to note that 3 of these study endpoints require the determination of absolute abundances, including endpoint 1, the decrease in bacterial vaginosis causative pathogens, endpoint 3, an increase in beneficial lactobacilli, and endpoint 4, the decrease in total bacterial vaginal load. The only endpoint that can also be established with relative abundance measurements is the reduction of vaginal dysbiosis in female patients. Since we know that absolute abundance measurements are important in the context of vaginal microbiome analysis, we partnered with another client of ours, whom I cannot name, but who is a world-leading developer of intimate care products, in order to establish the quantitative accuracy of PMP. To that end, we decided to use digital PCR as the quantitative gold standard. We selected a number of assays for microbial species that are critical for vaginal microbiome health, and we compared 97 measurements for serial dilutions of reference materials between digital PCR and PMP. Now, what you can see is a strong concordance as evidenced by a high correlation coefficient between both methods. In fact, the slope of the linear regression is almost 1, which would suggest a one-fold change difference, or in other words, no difference between PMP and digital PCR. Now, of course, pipetting errors are a reality and linear regressions can ignore outliers, which is why we plotted the distribution of fold change differences between PMP and digital PCR and reported the median. In this case, the median is 1.6, which is still below the theoretical minimum 2-fold change difference you'd expect to see when comparing different PCR methods. On the right-hand side, we have plotted the distribution of quantitative measurements returned by PMP versus dPCR, and we see a strong correlation. And if anything, the data suggest that PMP has a similarly strong quantitative performance at very, very low amounts of biomass than digital PCR. This performance held up across different assays and species evaluated, with continuously strong correlations, a median fold change below 2, and in this case a lower limit of quantification for PMP between 5 and 25 copies per microliter. Now, after having established the strong quantitative accuracy of PMP, I shall defer to Thermo Fisher's engineering prowess for helping us with accomplishing the cost and turnaround time requirements. The OpenArray architecture and the Applied Biosystems Quant Studio platform is for us very, very convenient, as it enables us to process samples from specimen all the way to report within several hours, and at a cost that is competitive and conducive to routine testing of the vaginal microbiome. So I hope having convinced you that the PMP technology is ideal for vaginal microbiome routine testing. And before concluding, I shall express my gratitude to our wonderful team at Biome, who have helped us establish a thriving test laboratory in Oslo, Norway, offering routine testing of gut, skin, and vaginal microbiome specimen, as well as custom assay development for microbiome test applications. With that, I would like to thank you for your attention and wish you all the best for your research.